Dear students, we are again going back into one more mode of selection of the transformed cells. Selection of the transformed cells is one of the sort of procedures or the techniques in genetic engineering or recombinant DNA technology. You understood that primarily in the last episode I was trying to tell to you about insertional inactivation, wherein if a foreign gene is inserted into a PBR322, the ampicillin gene or the tetracycline gene will get inactivated. Then it will primarily be detected when the colonies are grown on a sort of a nutrient medium which has got ampicillin resistance or tetracycline resistance. Now, in order to avoid, though that particular process is highly cumbersome, there is another sort of a process which primarily is termed as blue-white screening. Now, blue-white screening is based on the principle of insertional inactivation, and we are looking in terms of selection of transformations. So, selection of the transformants, that is the cells which have got the foreign gene of interest. How do you pick them up from bacterial colonies? So, it is primarily based on a technique of blue and white screening. Now, what happens in the blue and white screening? Normally, when you take Escherichia coli, E. coli, the bacteria has got its own DNA. The bacteria has got its own DNA and it also has a plasmid. This is the plasmid. The plasmid is the extra chromosomal genetic element. It has a gene called LAC-Z. LAC-Z is a gene which is present in the plasmid of E. coli. Now, primarily when you take LAC-Z gene, when you take the LAC-Z gene shows expression. That is, a gene is a component of the DNA, and the DNA is able to make the RNA and the proteins. So, and the protein is a sort of an enzyme. Thereby, the LAC-Z gene synthesizes, or it is responsible for the synthesis of beta-galactidase enzyme. This is, in simple, it is called as beta-gal. So, we have a LAC-Z gene. The LAC-Z is responsible for beta-galactidase activity. So, the expression of the gene, supposing a gene is expressed, it is seen in the form of an activity. And primarily in the activity we have there is high amount of synthesis of beta-gal in E. coli under normal conditions. Now, whenever we have the beta-gal enzyme, it has got a property. It is responsible for cleaving. Cleaving means breaking down. It is responsible for cleaving certain substrates, and it results in the formation of a colorless sort of a substrate called X-gal. So, we have LAC-Z responsible for beta-gal. Beta-gal in turn breaks down an intermediate compound which is termed as X-gal. It breaks down X-gal. So, because it breaks down X-gal is a colorless compound. So, when it gets broken down, the X-gal in turn synthesizes a substrate. It synthesizes a substrate. So, the X-gal synthesizes a sort of a substrate. You know the enzyme substrate relationship. So, thereby a substrate gets formed. The substrate which gets formed by the breakdown of X-gal, this substrate is a chromogenic compound. That is, it is easily colored. So, it is a chromogenic compound, and being a chromogenic compound, the substrate it goes to form a chromogenic compound. These compounds go to form blue colonies in a sort of a petri dish. So, in a petri dish you can easily mark the presence of blue colonies. So, what exactly are the blue colonies? The blue colonies are the chromogenic, they are the sort of colonies of Escherichia coli which are formed primarily due to X-gal. So, the X-gal substrates which are formed by beta-gal cleavation is due to the expression of LAC-Z gene in Escherichia coli, because Escherichia coli has got a plasmid which has got LAC-Z gene. Thereby, the presence of blue colonies indicate that it is the cells which are formed are termed as non-transformants. That is, they have not taken the sort of an insert of a foreign gene. So, normal cells with normal expression go to form blue colonies, and blue colonies do not have any particular sort of an insert. Now, we are looking at situation number 2. Imagine a situation wherein we have the lag gene, lag Z gene. We have broken down the lag Z gene by means of restriction enzymes. The lag Z is broken down. So, we are using restriction enzymes and we are cleaving the lag gene. And in the space which is got because of the cleaving of the lag Z gene, we insert the foreign gene of interest, or your recombinant DNA, is inserted in place of the beta gal. And once it is inserted, we understand that beta gal synthesis is blocked, because already it is inactivated. The beta gal is inactivated. It is inactivated and its synthesis is blocked. Thereby, because the beta gal synthesis is blocked and it is inactivated because of the insert of the recombinant DNA technology, there is no production of X gal. So, along with this, we understand there is no chromogenic substrate. There is no coloring substrate or no chromogenic substrate gets formed. Thereby, we do not have the formation of the blue colonies. So, instead of the blue colonies, we have the presence of white colonies. So, all the white colonies indicate that they are the sort of a transformants, or they contain the sort of a recombinant DNA. So, we have all white colonies which are given here in the form of X, X and X, and what do they indicate? They indicate the recombinants or the transformants with the foreign gene of interest. Thereby, this method of blue-green selection or blue-white selection, this method of blue and white selection primarily indicates that whenever we want to isolate the gene of interest, this is a fairly easy technique, because it is very easy to pull out and exactly study the growth of bacteria and also identify the bacteria under the microscope using a sort of a chromogenic substrate, whereby you can do the sort of a selection. Blue colonies indicate they are normal cells with no recombinants, and white colonies indicates that they have the transformants or they are the sort of a recombinants. So, this is an easier sort of a technique than probing and then identifying using two important antibiotic resistant genes like AMPR and tetracycline R, and again going for both for screening as well as for selection. So, this is a one-stop situation wherein blue-white screening is a very rapid method of detecting the sort of a selectants in biotechnology. So, whatever I have just told you in terms of blue and white selection relates to the previous class. Now, again as a sort of a link, we are now moving from biotechnology principles and process to BT and its applications. Between the sort of a link, you got to again be knowing about a very important sort of a vector which is termed as Agrobacterium tumefaciens.